All right, next year I'm going to make it shorter because that's it's a month. And I'll start my timer here so I don't go overboard. Okay. Thank you. Yeah, so thank you to the organizer for inviting me. It's always a pleasure to be here in Boston and meet with all of you, learning new things and also possibly providing some of my take on the problem of in-silico prediction of immunogenic peptide. And so, you know, that's my disclaimer. I'm, of course, an employee of Eli Lilly & Co. And, so following, you know, Jeremy's presentation obviously makes my life easier because I can fly through some of the initial slides and I don't have to repeat some of the concepts that he introduced today. Obviously, we are looking at the initiation of the adaptive immune response, which, let me see if I can get these, to work. Perfect, okay. And so obviously we're trying to identify what are the peptides that are responsible for priming and initiating the CD4 T cell response, which eventually will support humoral or cellular immunity. And something that is a little bit different from the concept of immunogenicity is the concept of immunodominance. And sometimes we do not focus on immunodominance and we fly directly to immunogenicity because I know that's what it's, you know, it's where the rubber meets the road, right? But the idea of immunodominance comes first to some extent, theoretically and biologically. So if you take a complex antigen, right, and you are trying to identify out of the different epitopes that you can generate from a complex antigen, how many will actually elicit a CD4 T cell response, and also that works for CD8, obviously. You find that the CD4 T cell response is primarily focused on a few of the many peptides that you can generate of a complex protein. The interesting thing is that on the other side, you have this idea of crypticity, right? That if you actually synthesize the peptides as a soluble K-mers, usually 50 mers or, you know, whatever length, and you have the peptide, you know, analyzed in a peptide binding study, you see that more many peptides will actually be able to bind to MHCs in vitro. And even if you take the same peptide and you throw them in a T cell culture, PBNC T cell, you will also see some responses of T cell to peptides that, however, you won't be able to generate if you just take the protein as a full protein, you let it process by your dendritic cell, and then you see what's the outcome. So there is a little bit of a difference between what you do when you actually take the peptide, and you do the 15-mer, you know, typical scanning versus taking the protein and see what actually happens. So, you know, the focus of research in the last, I would say, 3 decades, right, has been really the one of identify the immunodominant peptides for each protein, and obviously that, you know, serves many of the various streams of research that are pertaining to either vaccinology or immuno-oncology or immunogenicity in general sense. And so ideally we want to understand what happens, right, within an antigen-presenting cell when the decisions are made in terms of which peptide is actually generated through the processing of the full antigen and then is allowed to bind to the MHC and then is selected for presentation. And so if you think of what happens in, for instance, in dendritic cell, when the antigens are uptaken, they are undergoing the endosomal pathway, and through catepsins, they are cleaved onto smaller peptides. And then they are somehow allowed to replace the clip, which is a kind of a placeholder peptide for MHC class II molecules. And in the presence of a additional non-classical class II molecule known as HLA-DM, the clip is removed, and all these antigenic peptides can somehow attempt to bind the MHC, and then few of them will be selected for transport to the membrane and presentation to CD4 T cell. And so, you know, eventually we want to predict the outcome of the process. Now, obviously, this is a complex problem, and so we are trying to break it down into smaller, you know, pieces, whatever we can manage. So we go from epitope prediction into binding prediction, thinking that that is, you know, where most of the problem really lies. And then we called whatever binds an epitope, although we forget that the things are more complex than that. And so if you take a look at what was the state of the art in 2011, and we see what are the actual epitopes and what the different predictors available at the time could identify in terms of sensitivity. So by sensitivity, you're looking at the experimentally identified epitopes, and you see how many the virus predictor can actually pick correctly. You know that the most we were able to take about 55% of them. Instead, for the positive predictive value would be the other idea. If something is positive in my in-silico algorithm, how many of these positive are actually positive experimentally? So it's the other way, right? And so at the time, the best would perform are 10%. And that is the typical thing that we say in-silico prediction are largely over-predicting, right? So there is a tendency for positive predictive value. So, things got a little better, and most of the reason is that there has been an exponential increase in the number of peptides that have been measured in terms of affinity in peptide binding studies and then submitted to the immunoepitope database. So now that, the training data set has expanded enormously, although something that we discussed last year in this very context, there is a bias in terms of the MHC alleles for which this binding are actually performed. And so you have, for instance, HLA-DR1 or HLA-DR4, they have, preference in terms of binding experiment and the ensuing data set versus some of the most rare alleles for which the binding data are a little bit more scarce. But obviously now we are getting at very, large numbers. The other problem is, again, some of the predictions are based on, the theoretical foundation is not fully sound. The old way of doing prediction was fundamentally looking at the pockets that are lining the MHC binding site and just simply assuming that if the pockets are correctly filled by the side chains of the peptide, then the peptide will somehow be anchored to the MHC binding site and therefore will bind. So you do have some of the methods that are trained on the epitope data set and they are on the based on pocket profiling. They're not really performing well. I'll show you later some of the data. Other predictors are actually based on a matrix concept, whereby instead of looking only at the 4 pockets, you are basically considering the full nonamer that is responsible for the interaction of the peptide to the MHC binding site. And then by basically assigning weight to the different residue at the different position, these predictors are building real matrices. And so you basically have 20 rows and then 9 columns. And so you say that an alanine at position 1 has a much less likelihood to generate a favorable binding event as compared to a cysteine at position 7. And so you compile all these numbers and you basically have, you have your sequence of the peptide, you try to generate a final score of binding. Now this is a little bit better than the, the pocket profiling. The problem though is that we have found you cannot simply think that every residue provides a independent contribution to the binding of the peptide. And therefore just thinking of this as a simple table that you can just sum the numbers is not gonna work. This is one instance, experimental, evidence that the idea of just looking at the key in the lock model would not work. Some of my colleagues back in Milwaukee, they ran this very simple but elegant experiment whereby they took the HA306-319 peptide, which is a benchmark peptide for DR1 allele. And we know that it's an immunodominant peptide for H3N2 influenza in DR1 individuals. And then what they did is simply to hold the anchor position constant, but changing the other positions. And theoretically, even some of those that are supposed to be T-cell contact. And after applying 4 mutations at these positions that are basically either solvent exposed or are really pointing toward the TCR, they basically lost affinity of about 4 logs. So we are talking about 10 times less strong binding, although the so-called anchors were the same as the wild-type peptide. So that's definitely an evidence you cannot simply assume since you are fitting the pockets, then the peptide will be happy. So that's why when you look at the performance of the predictors, the ones that actually work better are those that are based on a neural network approach, whereby you're actually looking at the effect of a specific residue at a specific position in the context of the overall peptide sequence, looking at what the other amino acid at the other position can contribute or or decrease in terms of the binding. And so this was a paper from Andrea and al. From the IDB group, and published last year in Bioinformatics, where they did the benchmarking of all the various predictors, and they found that overall those that are based on neural networking are the best, with net MHC 2-pan being the one that performed the best, both in terms of the Spearman ranking coefficient or the AUC of the ROC curve. And then there was a even more recent publication from Zhao and Scher, and these are from Merck, and they have used an untested data set that they have generated in-house, and they have looked at some of these predictors, some of them the same as from the previous publication, and some of them are different. But overall, the idea is the same, that if you take the NN line, the net MHC 2-pan, the SMM line, and then the consensus from the IDB, which is basically a consensus, as the name says, of the scoring from some of these neural network-based algorithms, then that's where you have the best performing algorithms, whereas the epitope, the MHC flurry, and the comblib are in the second tier, if you wish, and then they show that in the ROC curve. So confirming what it was found last year. And then again, going back to the Andrea publication, they also tested how well they were performing in terms of identifying the actual T-cell epitope, not only the binding. And here you still see that even for alleles for which we have largest data sets, such as DR1 and DR4, when they had tried to see how well they were performing in terms of identifying the CD4 T cell epitopes, from the influenza, I think it was the H1N1 in this case. They obviously the most concerning aspect was the number of predicted but not found as experimental epitopes. So we are going back to the idea of having some false positives, right? But you also do have some false negatives here and there, which will be in this lower right quadrant. So in general, the HLA-2 peptide binding prediction platforms obviously can help with, some of the assessment, and but the problem is that they tend to overpredict. So they show the false positive, and also they are affected by a few false negatives. And the other problem is that binding prediction predicts binding. So we cannot really ask something that is supposed to do something, to do something else. So one has to account for the fact that the mechanism that generates a viable T-cell epitope is really much more than simple binding. And this was an interesting paper that was published by the Larry Stern group, and they are very close by at UMass here in Worcester. And so they basically look at, among the experimental, I would say, kinetic or binding aspect of the MHC2 peptide chemistry, which one could actually be the best proxy for immunodominance in the case of CD4 T cell response to the major core protein 8 and L from vaccinia virus. And so they look at the standard IC50. They look at, and they took the 800 nanomolar as their threshold for efficient binding. Now, keep in mind that things are a little bit variable here. If you look at IDB, By their ranking, the suggestion is that anything that passed the 5,000 nanomolar, so 5 micromolar of IC50, is a non-viable epitope. If you look at some of the Stern's lab work, they take between 800 or 1,000 nanomolar. Some of us, I've seen taking 500 nanomolar as another threshold, right, just to be more on the conservative side. So things are a little bit variable here. So they took the 800 nanomolar as the threshold of affinity, 45 hours as intrinsic half-life. So how long the peptide is bound to the MHC when there is no other factor involved in the release of a pre-bound peptide. The DM-mediated half-life. So now DM is that guy I mentioned before, somehow responsible for peptide editing and possibly identify what is the best candidate peptide to remain bound to the MHC and be transported to the membrane. And the presence of DM usually speeds up the release of the pre-bound peptide. So in that case, they took a 6 hour as a threshold for the half-life. And then the DM susceptibility, which is basically how much DM needs to have in reaction to get to a certain level of release of the peptide. And so overall, they found that the stability of a peptide in the presence of DM is the best predictor for CD4 T cell epitopes. And obviously, this is not something that we really account for in any of the algorithms, because generating a data set of DM mediated half-life is a pain in the neck to be, because you had to generate the MHC, you had to generate the peptides, and then you need to be able to generate enough pre-bound complex after purification of the unbound peptide you can actually do the reading of the stability of a peptide, and then you had to decide which platform you're going to use, and anisotropy-based or HPLC-based, and also that can be cumbersome. And you need to have DM, and it's very hard to have DM. Either you express it internally, or at this point I have not found anybody who can sell you DM like on the market, right? So you need to have your expressing cell line. So being able to actually generate a compelling data set to incorporate in silico prediction is very hard. So that's why either the MAPS based approach, or something that has been developed by the Southern Naxeri group in Hopkins, and it's basically a MAPS in a tube, if you wish, right? Are very useful in generating such a data set of peptides that are actually somehow, an informative data set that contains both the DM effect as well as the effect of cleavage. Because obviously this peptide, whether they're undergone the MAPS platform or they've been processed within your test tube, are subjected to cathepsin cleavage. So it's the best proxy that you can have to generating a viable CD4 T cell epitope. And now you can actually have, potentially creating a library that you can, incorporate in prediction. So that's, something that has been done recently by the IDB about a year ago. And I briefly mentioned this last year, but I want to expand this year on this because I think it's critical, right? So they have actually put together and curated a data set of 14,000 naturally processed ligands from MAPS that have been deposited within their literature search. And when they look at the ability of a ligand-based trained, so naturally processed ligand-trained data set, to pick new ligands, their performance was about 0.77. When they combined the cleavage and the binding, things got even better. So a even more recent and independent study that was done between Denmark, Argentina, and Australia, they have looked at both the IDB dataset in terms of affinity and ligands, as well as the in-house data that they have generated of naturally processed types. So think of a MAPS, right? So they ran their own MAPS on a certain number of antigens, and they have generated their own ligands. They have, they made sure that by doing all the right, assessment, that they would actually have the the curated library of peptides. They were assigned the alleles, and then they generate this final data set that I'm going to show you what it's made of, that contain both the affinity and the actual naturally processed ligands. And then they did both cross-validation as well external validation. So this is the overview of the data set that they have used for this study. The affinity data, obviously, the one that you get for the most part are based on 15 mers, right? Because that's what usually you synthesize, and that's what mostly the IDB, data set is made of, right? Because everybody is fundamentally generating 15 mers. Partially, there is a reason for that, because if you actually look at the eluted data, about 20% of the ligands that you elute are 15 mers. And then about 50% are between 14 and 16 mers, right? So you're about there in terms of length, right? So if you are a 15 mer, you're not going to miss, most of them, right? And obviously, you put your bang, you put your bang where most of your buck. No, you put your buck where is most of your bang. So if 15 mers work, right, why would they have to consider any other length, right? And then they considered a certain number of HLA2. Actually, they did both on, not only HLA2, MHC2 and MHC1 alleles. So they curated both, both sides of the coin. And some of the alleles were covered for about 100 ligands. Some of them were covered for about 10,000 ligands. So obviously there is again some disparity there, but more or less we're able to have a compelling, data set on which they could actually run some statistics. So when they looked at the performance of their prediction in terms of AUC of the ROC curve, for an IC50-based train algorithm to predict IC50, their performance again is somewhere around 0.72 or so. When they use the IC50 and the naturally processed ligands to predict IC50, their performance went up. When they tried to use only the naturally processed ligand to predict naturally processed ligands, their performance went up to 0.9 something. And then when they used both binding and ligand, meaning, again, elution from maps, to predict ligands, we are about, 0.95. So in a nutshell, if you are using both data set, the performance of your prediction will increase significantly. And the same story if you look at prediction in terms of percentile rank of a positive hit for a length-matched set of k-mers. And in this case, the smallest is this rank, the better is your performance, because now you're looking at where your positive hit is located in terms of the ranking distribution. So the smallest you are, obviously, the better your performance is. And so the similar thing was done for the external validation. So actually, they took data sets that were not curated in-house. These all were done outside. They look at both eluted ligands, and then they look at CD4 T cell epitopes as determined by tetramer staining. And so again, by looking at a predictor trained on affinity and naturally processed ligands, their performance was way better than the standard that's used nowadays, which is again net MHC 2, which is the 3.1 version nowadays, right? Much better in identifying ligands, a little better in identifying CD4 T cell epitopes, but still, it was a better performing algorithm. So we tried to do something very similar, comparable, if you wish, for some of the licensed molecules, licensed monoclonal antibodies, and we look at the, again, I'm stretching things a little bit. So I'm trying to, I wasn't a real fair comparison, let's say, because I'm trying to have this predictor tell me how well I can predict clinical immunogenicity. So it's not fair, right? Again, I'm trying to stretch things here a little bit, but at least as a proxy of, of responses, I sort of can have an idea of where I'm going here. So the IDB has a CD4 T cell immunogenicity prediction that puts together both the binding and possibly also what the T cell sees. The way they did it basically is that in a very unbiased way, they look at the series of known T cell epitopes this time, not binders, but epitopes. And so basically they didn't consider anything else than the pure sequence, irrespective of whether they would bind or not, what it was the allele, right? And so now their algorithm is trained to recognize patterns of similarities for which T cell might actually be activated for, and then you can couple that to the binding part. So you can ask the algorithm to run the combination, as they say here, the combine prediction on a 7 allele based binding set and what the T cell potentially might see, right? So when I've done that exercise and I said, okay, by looking at the score and then looking at the TADA from the inserts, how well can I look at that? So there is barely no correlation. But even if I try to set, let's say, 5% as my TADA for positive and negative, really I don't generate great numbers in terms of prediction. Okay, but I try to do a similar exercise as they did of combining also the naturally processed ligands and match what the prediction will tell me in terms of natural processing, along with the binding and what potentially the T cell can see. And so when you run that, there is another algorithm, that's the one I told about, within the IDB suite tools. And so you can actually put your sequence, and then they're going to tell you the score, the probability with which certain 30 mers, 40 mers, 50 mers, 60 mers, 70 mers will actually be potentially eluted. Exactly like a map, like a digital maps, if you wish. And so by doing that, you can actually see that those peptides are clustered. Actually, these are the total scores for the full sequence, right? And so what they did is that basically they took only the peptides that are from CDR, or anyway they have non-germline residue based on matching sequences, and then I sum the score. You can see almost you can cluster these these maps based on their cleavage score. And obviously, the highest the score, the highest is the probability this peptide will actually be shown off on the membrane. So when you combine the two on a peptide by peptide basis, what happens is that those scores for the cleavage now are pushing some of the positive into the negative realm. And by doing so now, you are actually increasing the number of the true negative. You are decreasing the number of the false positive, whereas the other one more or less remain the same. So still, one can discuss whether to put the threshold at 10 or at 5, but somehow a little bit the accuracy of the algorithm improves in terms of increasing the number of true positive and true negative, just because now I'm not only looking at the binding of what theoretically the CD4 T cells see, but potentially I'm trying to see whether there is a possible exposure of these peptides in a MAPS-like assay. So here the take-home message is that obviously HLA2 binding representation is a complex process with many factors. The predictors are focusing on the binding aspect of it, and the fact that we're having more and more platforms, whether they are true maps or data set generated through in vitro maps-like platforms, allows the generation of data sets that can inform better algorithms. So when you actually combine the two, the evidence seems to point to the possibility of better predictions. And obviously, in-silico, I don't have to inform this audience that in-silico is just a part of a much larger pipeline. So, that shouldn't be the only thing that you use. And now I want to acknowledge all my colleagues at Lilly in LBC in San Diego, and obviously ProImmune for the kind invitation. And then if you have any questions, I'll be happy to respond. Thank you.
That's your dog?
That's my dog. Yeah.
thank you for doing this work. So I think the other component of improving accuracy comes from what the T cells would look at. So after the binding, after improving the accuracy based on processing, then there's that whole component. So how would you incorporate that? Would you need to do similar effort with now the T cell repertoires, and what are the likely peptides that could bind on the outward facing pocket of MHC? So do you have any thoughts on that?
Yes. So so as I said, the IDB is using a if you wish, an agnostic approach to that problem. They have looked at what is in the repository indicated as a CD4 T cell epitope, right? And then without necessarily assigning any prior binding to MHC, they say, is there any amino acid pattern that somehow is comparable across all these known peptides? And based on that, I can try to sort of predict whether there will be a T cell able to see that. That comes with many caveats, and I don't think I have to list them here. The other thing is that, as we were discussing yesterday, some of the traits, such as hydrophobicity, has been identified a potential, character that along the peptide might actually generate a T cell response. Now, things are a bit a trick there, because it's true that if you look at the biophysics of the TCR binding to a peptide and MHC, and you have more hydrophobic plane, you are basically generating like a biochemical glue that facilitates any interaction between the TCR and the peptide MHC. And initially they thought that the binding of the T cell receptor to peptide MHC is fundamentally an entropic event, and so having hydrophobicity would increase that component of entropic interaction. Then you have example that's not the case, because if you take the HA peptide as a free lysine pointing upward, and those free lysines are generating salt bridges with the TCR of the known T cell receptor, and therefore that now this is not an hydrophobic based binding anymore. So it goes very much onto a situation by situation basis. What I can say is that taking a partition approach, one could do, you look at what's binding on one side, you look what is exposed on the other side, you try to make an inference from there, right? So that would be a potential way to look at the problem. Are any of the you look at the sequence of the peptide, you look at what potentially is visible from the T cell point of view by sort of looking at the positions that have been historically identified as T cell receptor contact, and you can make some inference based on that. Is not the most robust way, probably is an initial way to look at the problem. Also because there are so many indications that when the T cell receptor binds to the peptide MHC, it sees the whole thing, right? It's not simply that you have those 4 or 5 residue pointing up. To the extent that you can change a pocket position, and because of the changes in the overall structure of the peptide MHC, the same T cell receptor won't recognize it anymore, and it's just a pocket position. So now you have some of these from the Ross-John group or from Pipa-Marek. They have done so much in terms of cross-reactivity or lack thereof just by changing one of the residue pointing down. So, yeah, that part is very tricky because the variability that you have on the T cell receptor side of thing is, it's huge. So,
thanks very much, Andrea. That's really interesting. With the IDB class 2 cleavage prediction, you mentioned about the MHC assignment following the MAPS data. How do they do the MHC assignment? Because I guess that's fairly critical with.
That's correct. So in the natural processing, there is no assignment. There is no assignment when you look at really at the output of the predictor for natural ligand.
Yeah.
You basically have the sequence, right? You input the sequence, and then they give you a list of peptides that are divided by, you can sort them by length,
Yeah.
by score, And it's basically a prediction of, what is the likelihood that on a random dendritic cell, you're going to see that peptide being displayed.
But on your table there was the assignment was the next step in the process. So they were assigning which MHC that particular peptide was going to be.
That's not in a natural, that's in the CD4 T cell immunogenicity, which
Yes.
is a different thing.
So
in that thing, you have, what you can do is that you can either select the immunogenicity based on binding. So they do the 7 allele, which is fundamentally a clustering of the MHC into superfamilies, right? And so they say, okay, we'll give you the immunogenicity only based on peptide binding. Then you can decide only on the T cell based on this assumption that they construct this pattern of recognition, or the IDB recommended is a combination of the two. Yeah.
I have a question which is a little bit unrelated, but since you are an MHC class 2 processing pathway expert, I'm going to ask you that question because I could not find any papers that would describe this. Do we know if the clip peptide ever goes at the surface? Is it processed? Have you found it in MAPS assay? And do we have T cells that can be activated, or are we tolerized to clip? The two goes together.
So so the clip peptide is actually presentable, and we know that from the study of the H2M knockout mice, that basically is the murin DM. When you take away the M from the scene, most of the things you see on the surface are empty MHC or MHC bound to clip. And then all the CD4 T cell response is completely blown because now you can't train on anything, right? So you can have peptide bind, you can have clip bound to the R, and in few cases you can elute it off the MHC even in a DM competent, but you remember that the binding of the clip to MHC alleles is variable in terms of affinity. So which is why for certain alleles you need to have DM for the removal. Because, for instance, that's a case of HLA-DR1. There are certain alleles that the binding is so, low affinity that even in the absence of DM, you can still have the exchange. It self-removes in the endosome. So it's on a allele basis, and it's on, on the presence of absence of DM. But so because in the TEC, thymic epithelial cells, DM is inhibited by HLA-DO. Now, the likelihood that the clip bound to an MHC will be presented to a maturing thymocyte is very high because you don't have DM doing that exchange action. And so, I think that you're basically going to have negative selection of the thymocytes that can see the clip just because it's expressed there. All right. Thank you.